adding spores to a gut microbiome in 3 weeks increased the diversity of the gut microbiome by almost 25 to 30%. And it increased the growth of keystone species, and it brought down pathogens. So the spores are like the police or the orchestrator of the microbiome. The point of fermented foods is not to deliver bacteria to your system, because those bacteria are designed to ferment the food outside of the body. The moment you eat them, the stomach acid is going to kill all those bacteria. So fermented foods are foods that now contain nutrients that the food naturally did not contain, and those new nutrients come about from the fermentation process. Natural, actual made kombucha using traditional techniques can be very beneficial for you, right? Because they have all these amazing organic acids in them. But store-bought kombucha in the bottle is not even kombucha. All these companies started competing with each other on this billions of CFUs idea. They started creating this false notion that the potency and efficacy of a probiotic is dependent on its CFU count, when it actually is not.
Kiran, what would you say an optimal microbiome looks like? And why is that essential when it comes to our health?
That's a million, maybe $1 billion question. But when you look at the tens of thousands of research studies that look at the health and function of the microbiome, there's one theme that seems to be present across the board, and that one is that high diversity in the gut microbiome is absolutely paramount to health, right? So one of the best features of a healthy microbiome is that it's high in diversity. Now, there's a number of reasons why that provides functionality, protection, resilience, and so on. So when we think about it, number one, let's go to our genes, right? Genes dictate a lot of functionality within the body. Now, we used to think that virtually every disease and disease process had a gene associated with it. And so that was part of the goal of the Human Genome Project, was to sequence the entire human genome, and we'd find the heart disease gene, and we'd find the cancerous gene, and we'd find the Alzheimer's gene, and maybe we can tweak the expression of those genes in order to alter the progress of disease. But as it turns out, when they sequence the human genome, what they predicted was that we would have somewhere in the order of 250,000 genes. As it turns out, we had about 1/10 of that in terms of functional genes, right? We have about 22,000 functional genes. So that was a big mystery, because an earthworm has 30,000 functional genes, right? So how is it that we're so sophisticated? And how is it we're at the top of the food chain and evolutionary ladder? And that was part of what spawned the Human Microbiome Project, because then the assumption was maybe we're getting genetic help from somewhere else. Maybe it's the microbes that live in and on our system. So then when you look at the microbes in the gut alone, the microbes in the body provide over 2 million genes that are functional, right? So we are far more, 150 times more genetic from a functional standpoint, from the microbes versus the human itself, right? So a lot of our capabilities to function as a human comes from the microbial ecosystem and the genes that they provide us, the capabilities that they provide us. Now, of course, the more of them there are, the more diversity there are, the more genetic capability we have, right? So when we lose diversity, we literally lose function. And we cease to function as a human should function. So that's one reason why diversity is so important. The second part of it is a diverse environment is resistant to invasion, right? So people with very diverse microbiomes tend to be resistant to infections and pathogenic invasions from bacteria, viruses, fungi, and so on, because diverse communities are much more stable. They have a lot more competition. They can fight off an incoming, invading pathogen. And so people that tend to have diverse microbiomes tend to have a much more resilience, and their immune systems, and then thereby their ecosystems, can also deal with exposure to lots of things that we all get exposed to all the time. Then the third part has to do with the immune system as well. The immune system is largely dependent on the microbiome for functionality and for intelligence, to truly understand how what the environment looks like around that human, what to attack, what not to attack. A diverse microbiome does a better job of communicating and tutoring the immune system. What studies show is that as you lose diversity in the microbiome, you start to have more and more dysfunction in the immune system. The immune system is losing its intelligence and it's losing its capability to understand the environment around it. And so it just starts attacking everything it comes across, right? So that's another reason. A fourth reason I would put out is just the ability to break down, digest, and assimilate nutrients, right? We count on our microbes a lot to break down lots of different types of foods. We know, for example, fiber is really important. Fiber is arguably one of the most well-studied macronutrients there is. There's lots and lots of studies. In fact, a large meta-analysis study just came out on almost 1 million patients on fiber intake. And the study was very clear that when you add fiber, you increase longevity, you protect yourself against disease. And in fact, for every 10 grams of fiber that you add into your daily diet, you reduce your disease and mortality risk by 10% to a certain degree, right? And so why is it that fiber is so important? Well, we don't break down fiber. We don't get any nutrition from fiber directly, right? We can't even digest it. It's because it goes to the large bowel where it feeds the big diverse set of microbes, and it increases diversity. Microbes then convert the fiber into very usable compounds. Things like, urolithins converts the fiber to short chain fatty acids like butyrate and so on. So that breakdown, assimilation, and utilization of nutrients is another important part. So we just keep those four things in mind, right? The genetic and the functional capabilities that our diverse microbiome provides, the resilience to infection, the importance of the communication of a diverse microbiome to the immune system to teach the immune system about the environment that's in, and number four is the utilization, assimilation of nutrients from food sources. So just those alone makes all the difference in your overall health outcomes and your resilience as well.
All right, so you've stressed the importance of the diversity. When we're talking about diversity, how many different types of microbes are there in a healthy gut?
In a healthy gut. Now, when we look at a healthy Western gut, there's some marked differences compared to, like, let's say, a healthy sub-Saharan desert African gut, right? People that are foragers that eat lots of roots and tubers and all that. So we did, when I was at Microbiome Labs, we did, probably in my time, 15, 20,000 microbiome tests on individuals in North America. And then we were looking at tests from Papua New Guinea tribes, Hadza tribe in Tanzania. The average diversity that we see in North America, in terms of number of viable species in the gut microbiome, was around 130. Right? Now you compare that to some of the hunter-gatherer tribes that represent what our microbiomes used to look like prior to industrialization and commercialization of lots of things that we consume and so on. They tend to have 600, 700, 800 species, up to 1,000 species. Right. So we can be 20% of where we need to be as a species in terms of the number of viable microbial species that exist within our system. So I would say a healthy individual in the Western world, if they are at 250 to 300 species or more, they're doing an amazing job, right? Because they probably didn't get passed down enough organisms to begin with. Now, if you're in sub-Saharan Africa, you could be upwards of 600 to 1,000 species. So if you are below 200, you will likely have some compromise to those four things that I talked about.
Okay, so again, we've emphasized the diversity piece. How much does volume matter? So there's two separate pieces here. We can have a number of different microbes, but then how much of them do we need for optimal gut health?
So the interesting thing about microbial ecosystems, right, is that microbes will utilize all of the real estate, no matter what the diversity looks like, right? So any square millimeter or nanometer where a microbe can exist, one will exist. And so if you have, for example, let's say, just use a number that's been thrown out. Let's say you have 40 trillion microbes in your system, which is some of the latest estimates of the number of microbes that exist within the human system. 40 trillion. That 40 trillion can be represented by 150 or 200 species, or it can be represented by 100 species. That 40 trillion number rarely changes, right? Because anywhere there's open real estate, microbes will fill that space. right? So, and that's part of the problem as well, right? Because what can happen easily is that if you make certain lifestyle choices, and let's say there's a great community of very important microbes right in this given region, and you make a lifestyle choice that knocks them down quite a bit, that spot will be taken over pretty quickly, within hours sometimes, by other competitive microbes, right? And if that keeps happening, then what happens is you get certain competitive microbes that do really well in taking over territory, starts taking over too much territory, and diversity goes down, and concentrations of certain microbes go up. And when concentrations of certain microbes go up, they become less helpful and more problematic, right? So pathogens are a good example of that. Everyone has pathogens within their microbiome, right? And many of these pathogens actually do have function within the microbiome. But like any sort of potential toxin, the dose really dictates the poison, right? In this case, the level and the concentration of that particular pathogenic species is what dictates whether it acts as a pathogen or it doesn't. Right. So you could have, a type of Clostridia in there that could have pathogenic potential, but it could actually be doing useful things in your proximal large bowel, like, breaking down bile acids or breaking down hydrogen sulfide and so on. So it's reducing the inflammation in the gut. The moment those microbes are allowed to overgrow and take up more territory than they should, they go, hey, guys, this is a chance for us to take over the system. We are opportunistic pathogens. This is an opportunity. So they start turning on their virulence factors and then start to become more foe than friend, right? So that can happen quite easily in the microbiome. You've got a lot of opportunistic pathogens that live there naturally. If you give them the opportunity, a lot of times it's more real estate, more concentration. They will take that opportunity to try to take over the system.
So what I'm hearing you say is it's more about the balance of the microbiome versus good guys and bad guys, which would be typically how allopathic medicine would look at the gut.
That's 100% clear, true. So if you have this notion of good versus bad, then you start coming up with things like, well, if something's wrong with the microbiome, let's just kill the bad, right? And reseed with the good. Well, it doesn't work that way, right? Because you can't just specifically go in and kill ones that have the potential of being bad. And in fact, the act of killing in itself could create enough disruption to the ecosystem where it allows the opportunistic organisms to come back in a more unfavorable way and get to levels that actually causes them to become virulent, right? So we tend to have this component of our microbiome that's called the pathobionts. So these are called pathobionts, and the biont part is important because as a biont, it means they live naturally within us, within the organism, right? But they are pathobionts because they have pathogen capabilities. Now, if you have high levels of pathobionts compared to non-pathobionts, you will have opportunities where these pathobionts start to rear their ugly heads and start to elicit virulence factors, right? So we saw this, for example, in COVID. There was a lot of really amazing work done by Liam Mahoney out of the University College Cork in terms of mapping the microbiomes of people that had very severe infections and even ended up dying from COVID or got hospitalized. And then he was looking at the connection between what their microbiomes looked like and how they responded to exposure to this new virus, right? And what he found is that if you had a certain amount of pathobionts, it was very predictive of a very negative outcome from exposure to this virus. In fact, once they built the model out, he could start looking at cases, just looking at microbiome analysis without knowing anything about how the patient fared. And he could predict whether that patient got hospitalized or went as far as succumbing to the infection based on what the pathobiont mapping looked like, right? So what that shows is that if you have this high level of opportunistic pathogens relative to non-opportunistic pathogens, relative to what we may call as commensals, then given the right opportunity, those can go haywire. And in this case, the opportunity was a new viral infection, all the over-sterilization, all the stress, all the things that are associated with it.
If you're enjoying the episode, let me know by clicking like and subscribe below. Thank you so much, and now back to the show. As you talk about the virus there, it gets me thinking about different pieces of the microbiome, because typically, in a general sense, when people refer to the microbiome, they're talking about bacteria. And as we talk about microbes in the microbiome, people are assuming we're talking about bacteria, but there's more than that going on there. So we know now the diversity piece, the volume piece. Let's talk about the different types of microbes that are in there.
Yeah, and so you're right. So it's not just bacteria; it is majority bacteria, but there's viruses, there's fungi, there are protozoa, there are amoebas, there are all kinds of things in there. Of course, many people have parasites that naturally live with them as well. That could be a normal part of their microbiome, depending on where you grew up and where you lived. And so the diversity in terms of the types of organism, not only within the bacterial kingdom, you'll find really high diversity in terms of the types of organisms. Now, talking about bacteria, because they're the most functional within the microbiome, right? They're the ones that we know the most about in terms of how they affect our health and function. So when we talk about the different groups of microbes, the ones to really keep in mind that have been shown over and over again to be critically important to health outcomes, disease resilience, and disease risk are what we would categorize as keystone species. Right. So keystone species are called that because, like in a given ecosystem, anytime you have keystone organisms, their function in that ecosystem means that function is critical to the maintenance and stability of that ecosystem. Right. In some cases, in other ecosystems, not the microbiome, it could be an apex predator. Right. An apex predator controls the population of certain prey. That prey can then has a control over the population of certain fauna, and that fauna maintains diversity. So, like, you lose the apex predator, all of a sudden its prey is overpopulated, the fauna gets eaten out too much, and then the whole system starts to fall apart. Now, the same thing happens in the gut, and the rest of the microbiomes in the body is where if you have a keystone organism, the organism's function maintains the stability of that ecosystem. Now, in the case of the gut microbiome, you've got numerous microbes that have been identified as keystone organisms. For example, Faecalibacter prosnitzi, Akkermansia muciniphila, Bifidobacterium longum, Bifidobacterium animalis. These are very important microbes to the overall health and balance of the microbiome. And in the case of keystone species in humans, we're also seeing that these microbes are protective against categories of disease, right? So you take Faecalibacter prosnitzi, for example. When Faecalibacter prosnitzi is at healthy levels within the system, you are protected against inflammatory conditions in the bowel. Things like IBD, Crohn's, colitis, microcolitis, even colorectal cancer to a certain degree. Akkermansia muciniphila, right? So Akkermansia is inversely correlated with metabolic syndrome and all the diseases that go under it, like obesity, diabetes, cardiovascular disease, even things like polycystic ovarian syndrome and so on, because when you have good levels of Akkermansia, it provides protection against those disease pathologies. We'll talk about how it does that, but it's important to note that when we look at diversity, we want uniformity within the number of species, but we also want good, stable levels of these keystone organisms. And that's a very important part of the health of a healthy microbiome.
Somebody coming to this conversation saying to themselves, okay, I'm already feeling overwhelmed. There's so many different species. There's a lot to this. And it sounds like if I've been, born over here in the West and grew up over here, I'm going to be lacking in a big way. So in a practical sense for that person, what's the first step? Is getting some kind of testing done to determine where we're at on the spectrum with our microbiome the first step, or how do we begin?
Yeah. So step one to me is incorporating into your life behaviors and choices that will always increase diversity and increase keystone species, right? So if you're thinking about, oh my God, my microbiome is probably devastated. I've had multiple rounds of antibiotics in my life. Maybe I was born C-section. I grew up in with parents who used a lot of over-sterilization in the household, right? We did a lot of things that we know now to be harmful to the microbiome that we didn't know was harmful before. So then you're starting to think, okay, how do I fix this? How do I improve my microbiome? I would have them focus on two outcomes. The two outcomes are increased diversity, increased keystone species, right? So here's how you do that. And it's the beauty of it is it's relatively simple. So to increase diversity, some of the things you can do right off the bat are to increase the diversity of the foods you eat, right? The higher the diversity in your diet, the higher the diversity in your gut microbiome. And the reason for that is because different microbes feed on different macronutrients at different rates, right? So you might take, a root, vegetable, like let's say you take a sweet potato, or you take a different form of a carbohydrate. Let's say you take a pineapple. They have very different carbohydrate structures in one case a vegetable, one case of fruit, or pineapple is actually technically kind of like a seed of some sort, right? And so if you take those two food items, they have very different carbohydrates in them. They will feed different classes of microbes, which require different enzymes to break down the different structures in those carbohydrates. You take lean meat, for example, there's going to be different microbes that are going to break that down. Take nuts and seeds and fibers, soluble fibers versus insoluble fibers. You're going to get different microbes that have different capabilities of breaking those down. So the more diverse your diet is, the more diverse your microbiome is going to be. If you just think about like our ancestors, there were some anthropological studies that estimated that early humans consumed upwards of 600 different types of foods annually, right? At best, a Westerner today maybe consumes 15 different types of foods, right? And you just journal it yourself. Take a week's sampling of foods and just look at how many different types of foods you consume. And it's not that much, right? So that's another thing that's driving a lower diversity. So increase the diversity of your diet. That's one. Number two, you can add things like intermittent fasting. Fasting, and it's counterintuitive, actually does increase the diversity of your microbiome. And part of the reason is because when you look at how microbes in the microbiome break down food products, you've got primary digesters, secondary digesters, tertiary digesters, right? The primary digesters will take in the main food that you're consuming, start breaking down the big macromolecules, and then they'll create byproducts from that breakdown. Then there's a whole set of microbes that consume the byproducts. And then they break those down and create new byproducts. And then there's another set of microbes that break down that set of byproducts, right? So each time there's a cycle going through and a stage gating of microbes that become really active because of what the substrate that is available at that time is. Now, one of the reasons why fasting can be very beneficial is if you take in a meal, right? You take a bunch of protein, carbohydrates, fats, and all that, all the primary digesters are now going to work, right? And it's going to take them a while. It's going to take them several hours to start breaking that down. And then once they're done with their work, the secondary digesters go in and they start breaking down the byproducts. But if in that time, more macromolecules come in, more food comes in, the primary digesters are the ones that are going to work most of the time, right? They're doing the first stage of digestion. So the secondary and tertiary guys aren't really getting enough activity. They're not getting enough substrates. They're not getting enough time to start proliferating and do what they're supposed to do. So that timing issue is really important, right? So if you can add in 12, 13, 14 hours in a day, mostly overnight where you're not eating anything, not consuming any foods, that can help the diversity as well. The other thing to think about with diversity is getting outside, being exposed to the environment, the outside environment, right? Especially natural environment. So if you have natural areas around you, around me, I have a lot of forest preserves to go on hikes and mountain bike rides. If you're on the coast, you can go to the beach, you can go walking along the shore. All of those things increase the diversity of your microbiome because you're engaging in nature. And one of the things I tell people to really enhance that, if you want to enhance the effect of nature on your microbiome, one of the things you can do is just be very deliberate as you're walking through, say you're going on a hike and on a trail, picking up things and touching things, right? Touching leaves, touching barks, picking up rocks, and doing that kind of thing. So physically engaging with nature and then sitting down and eating in nature. That is one of the most natural things because we all sat down on the dirt and ate. Our ancestors did, at least, for the most part of human evolution. And if you look at most of these hunter-gatherer tribes and all that, one of the ways in which they all eat, they all sit around in a circle on the ground and they eat, right? And so you're really engaging with nature and gaining lots of exposure to good microbes outside that will stimulate diversity within your gut microbiome. A fourth thing that you can do is also bring in a pet, right? So dogs, for example, studies have shown households with dogs tend to have people with higher diversity in their gut microbiome because the dogs go out, they bring in a bunch of organisms and they put them all over your house, right? So engaging with that dog, having pets in the household does increase your diversity. Then the last thing to think about, and maybe two things to think about at the end, are not over sterilizing your home, right? So don't use, antimicrobial cleaners on every surface of your home. If you want to antimicrobial your toilet from time to time, fine, or the shower, maybe there's mildew growing, you can do that. But most surfaces in the home does not need to be sterilized, right? Like my desk and dressers and all of that stuff, we never sterilize it. Right. The kitchen counter from time to time, if we bring home some raw chicken and raw chicken juice gets on the counter, you do want to sterilize that because you want to get rid of any salmonella that might be on it. But most surfaces in your home do not have to be sterilized. We over sterilize our environment. So you need to build diversity in your home biome in order to support your own diversity in your gut microbiome. Right. And as much as you can, keep the windows and doors and all that open so you expose outside microbes into your internal environment. The last thing that I was mentioning is managing stress. Stress is one of those things that actually reduces diversity. And we all have stress every single day, right? So things that you can do to manage or reduce the impact of stress on your body becomes really, really important in terms of maintaining diversity in the gut microbiome. So those are all the things you can do to add into your lifestyle that cumulatively will have a huge impact on your gut microbiome. Now, how do you specifically increase the keystone species, right, within the microbiome? All those things I talked about will just increase lots of different organisms. These couple things will specifically increase keystone species. Number one, high intake of fiber, right? Mostly soluble fiber, some insoluble fiber. But there's really no upper limit to fiber intake, right? So I shoot for close to 100 grams a day of fiber from a variety of sources, mostly from food. I do some supplementation as well, just even simple things like psyllium husk, right? You can get an additional 15 to 20 grams if you just make a drink out of it. But you can, and chia seeds and a number of different things that you can add in there. But I try to get it mostly from food as much as I can, and I try to calculate that I'm getting somewhere close to 100 grams of fiber a day. That is critically important to feeding the keystone species. Now, on top of fiber, you need prebiotics, and prebiotics are different than fiber. Fiber is kind of roughage, right? Prebiotics are normally oligosaccharides or inulin. So think about fructooligosaccharides or galacto-oligosaccharides or xylo-oligosaccharides. These are typically taken in supplement form, and it's good to do it in supplement form because it's easier to ensure that you're getting the right amount of it, right? So you want somewhere between 3 and 5 grams a day of these oligosaccharides. These oligosaccharides specifically feed keystone species. Most of these keystone species are the only ones that have the capability of breaking down and consuming these oligosaccharides, right? So fiber, lots of it soluble, oligosaccharides. And the third thing is polyphenols. Polyphenols are really important. Polyphenols, of course, come from lots of red and colored fruits and vegetables. Polyphenols act as a potent prebiotic that feed certain types of keystone organisms within your system. So, right? So that's a critical aspect of getting some of the keystone species to increase in their growth as well. So the natural food sources of prebiotics will be things like eating the husk of certain fruits. Or eating the internal parts of the fruits, right? The fibrous components of fruit. So if you think about pineapples, mangoes, kiwis, they will all have some degree of oligosaccharide prebiotics in the husk part, on the fibrous part of the fruits. Now, a lot of sources of fiber as well, things like seeds will have some oligosaccharides in there. Mushrooms actually have unique oligosaccharides in there. So you do get them from food. The thing is, it's hard to discern and know that you're getting the right amount, right? With oligosaccharides, dosing is important. Most studies show that there's a certain threshold dose where once you get to that dose and go above, you're really getting the benefits of the oligosaccharides, right? Fructo oligosaccharides are the ones that come from fruits. Xylo oligosaccharides come from certain vegetables. In the case of supplements, the xylo oligosaccharides actually come from the inner part of the corn cob, which nobody really eats that. And so those kind of roughage, and like, pigs would eat the corn cob, humans just don't, right? And so we throw out a lot of the very fibrous, kind of gritty parts of fruits and vegetables because we don't like that part, but that's where a lot of the oligosaccharides are. Galacto oligosaccharides are ones that come from dairy. And a lot of people avoid dairy anyway. You have to be consuming the full fat dairy in order to pull the galacto-oligosaccharides into the mix. So that's why I say prebiotics, I like to supplement with it because I know I'm getting some from the foods I eat, but it's just really hard to understand if you're getting the right amount. And they're really important, right? So I want to try to get 4 to 5 grams minimum a day, in addition to the fiber that I'm getting, which is separate. Now, also think about prebiotics and fiber functioning a little bit differently in the body. So prebiotics are very specific to the types of bacteria, down to the species that they feed. Fiber can feed lots of different bacteria, right? There's lots of microbes that can utilize fiber in your system, but very few that can utilize these specialized prebiotics. Prebiotics tend to have much more complicated structures, carbohydrate structures, than fiber does.
You mentioned the prebiotics, they typically feed the keystone species. And it sounds like fiber feeds the whole spectrum, including the keystone species.
Including keystone, yep. And utilization, and here's a tip about fiber, right? I mentioned that I try to get upwards of 100 grams of fiber a day. Now, if you're someone that's just eating a regular Western diet, you don't want to have 100 grams of fiber tomorrow, right? Your system's going to go into some sort of shock. And you won't even be able to utilize all of that fiber because it takes a little bit of time to start to train your microbiome to utilize the fiber because your microbiome adapts to your diet, right? So if you've had a diet that's largely protein or largely certain types of simple carbohydrates, or maybe you've been doing keto for a while, so you're getting a lot of fat, and now you go, okay, I want to incorporate fiber, you need to bring up the population of all of those microbes that can utilize the fiber, and you need to train them a little bit. So my recommendation for fiber is usually start with a goal of around 10 to 15 grams a day, right? And then each week bump it up by a couple of grams and just get the system ready for it. Oligosaccharides, you can go 1 or 2 grams to begin with if you've never really taken it as a supplementation. Start with a couple of grams, 1 to 2, and then add in 1 to 2 grams a week till you get to that 4 or 5 gram mark. Right, so sort of bring both of those in slowly, or you won't utilize it effectively, and you may get a lot of gas and bloating and things that are making you uncomfortable, which means that people might stop. Right, and that's one of the most common things that occur with fiber is that people start to increase the fiber intake, they get bloaty, they get gaseous because the wrong microbes are utilizing it in the wrong way, and it takes a little bit of time to adapt the system to actually utilizing the fiber properly.
Coming back to a few minutes ago, you mentioned the benefit of having a diverse array of different foods for the microbiome. It sounded like that was predominantly because we want to give the microbiome the food it needs. And you mentioned this hierarchy where the microbes at the top are going to break it down for some of the microbes below. I'm curious, though, when we consume these different foods from different parts of the world, if we're going to a grocery store and getting all different and a diverse array here, how much bacteria do we get on the produce itself that's getting into the system versus giving food for what we already have?
Yeah, unfortunately, our food is relatively dead now. It used to be that we got huge number of organisms in with our food, right? We consumed non-sterilized food for the most part. And then, of course, we started preserving food by fermenting it, and then you're getting lots of microbial exposure to the fermented versions of the food. And so our answers would eat things with dirt on it, and we just don't do that anymore. Know that in the U.S., when most produce and vegetables and all that are harvested, they all are put through a chlorine wash, right? And so they're sterilized to a certain degree. Now, the level of chlorine is not going to sterilize the food, but you're knocking down the microbes quite a bit. So we rarely get any sort of beneficial microbes on our produce. This is why it's also important, if people can, to try to grow some of their own food if possible, right? Having a garden, and not everyone's going to become a farmer and start growing all their needs. But let's say you've got a tomato garden, or you've got tomatoes and cucumbers. Maybe you've got tomatoes, cucumbers, and one other thing that you're growing. Even just adding those, and you're using good, clean soil, I would caution people against going to your hardware store and buying a bag of soil, right? Because some of that soil is treated quite a bit with lots of chemicals and all that to add growth factors and all that. What I would actually do is utilize any soil that you have access to. In our case, when we built a garden at my house, we just used the soil that was there on the dirt underneath the grass. The problem is we actually went as far as looking at it under a microscope, and we saw that the soil was virtually dead. Right? There's like no microbes in that soil. And of course, not surprising, because people have been using glyphosate and all these pesticides and herbicides and all that on the ground for decades now. So those things tend to sterilize the environment. So what we did is we actually got some fermented peat moss, and we got some rock dust. So you can go to the quarry, a local quarry, and get some rock dust. And just raw rock dust just from the quarry itself. And then we actually went out on hikes and just grabbed soil from the natural environments, right? Around the trees and where there's a lot of vegetation and shrubbery and all that, that soil is teeming with microbes. And then we got that, I mentioned the fermented peat moss. So those few things that we added and brought to our soil and mixed it in, brought life back to the soil. Right. So that adds microbes to the environment and adds it to it. Now, we use spores as well, because we were using in our factory in India, when we were manufacturing the spores, we were actually taking the ferment. Once we isolated the spores, we were taking the ferment from the spores, and we were spreading it all over the area around the factory where we had a green space, and it looked like a rainforest. Because it was such great food for the soil and for the microbes. So I started doing the same thing. I started fermenting with spores at home, and then pouring the ferment into the soil as well. So we had an amazing garden at my previous house. We haven't built one here yet, which we just moved to. We had an amazing garden that looked like a rainforest, right? And we were getting cucumbers, jalapenos, tomatoes. I think we got carrots out of it. And a number of things. Now, when you eat that kind of food and the soil is healthy, clean, doesn't have chemicals, you've brought life to the soil, you can eat that stuff without sterilizing it, right? We would pluck the cucumbers and maybe rinse it off a little bit with just regular water, and then cut it up and eat it. You add that to your diet, you're bringing in living food into the diet. Maybe 60%, 70% of what you're eating came from the grocery store, which is fine. You're at least bringing in living food to add to it.
You mentioned this chlorine bath that our produce at the grocery store is getting. Do you know if that's on organic as well?
It likely is to a certain degree, yeah. Because I think that is more of a food safety issue. Believe it or not, produce is a bigger source of E. coli infection than protein is, right? The most common is things like lettuce. And cabbage and so on. And in fact, tomatoes are a big source of it. So Campylobacter, E. coli, and all that. So, and part of that is because our soil is so unhealthy and contaminated. This runoff from this factory farm next door where the animals are being hit with antibiotics, right? And because they're hit with antibiotics so much, their pathogen loads tend to be really high because their microbiomes are all off, right? And that provides the opportunity for the pathogens to go. So the amount of E. coli that our cows shed, the amount of salmonella that our chickens shed are way more than what the natural grazing versions of those animals would, right? And so that runoff and all that also contaminates the soil. And I used to work in food safety as a microbiologist, and we would do experiments where you plant tomatoes with salmonella contaminated in the soil. The salmonella will actually end up going through the roots and into the tomato. So you could have a tomato now that looks perfectly clean. You could even clean the outside, but you bite into it or you cut into it, you've got salmonella on the inside of it, right? So that is a big danger in the food industry, in the industrial complex that we have for the food system. So they chlorine wash everything as part of the food safety protocol.
You mentioned pesticides and herbicides, whether it be conventional, which is typically going to have more of those on it, or organic if there's cross-contamination. Talk about what we can do to help bring that load down, whether it be washing in something specific or just using water. You can let us know what's best. And then talk about the impact of those on the microbiome if they get through.
Yeah. So let me talk about the impact first, right? So there's a lot of studies that show that Roundup, for example, and then that's the most prevalent pesticide, herbicide that's being used. So we'll focus on that a little bit. Roundup, which the active ingredient is called glyphosate, and glyphosate is an antimicrobial. To give people an understanding of how it works, it interferes with something called the shikimate pathway. The shikimate pathway is a way in which microbes, or in this case, plants, use, so weeds, for example, and insects and all that. They use this pathway to create aromatic amino acids, which is an important part of metabolism and normal function. And so this compound interferes with that pathway, which means that the weed or the insect can't function anymore, and it kills them off. And so the premise around the safety of that compound was that, well, humans don't have the shikimate pathway, so it doesn't have an effect on us, right? And so that was the premise around the safety. Well, the problem is all our microbes in our gut have the shikimate pathway and require that in order to produce their aromatic amino acids and function in the right way. And so what we're starting to see is that although it's not toxic to the human cell, it is toxigenic, and it is a bactericide to the microbes in the gut, right? And it's a bactericide or antibiotic in one of the worst ways because it actually selectively kills the good bacteria. There's lots of pathogens in the gut, like Pseudomonas and certain types of Clostridia and all that have figured out ways around this compound, so they're resistant to it. And lots of your lactic acid bacteria, your beneficial commensals, are very susceptible to it. So when you get exposure to it through the food system, what's happening is you're getting small doses every day of an antibiotic that specifically kills the beneficial bacteria, right? That is a perfect opportunity for the pathogens to overgrow and take over, right? So this becomes a definitive component of your ecosystem. Now, what is the impact of that, right? We did a study with King's College in London because we were concerned and we were interested to see what does it look like in a pediatric microbiome to get exposed to cereal level of glyphosate each day, right? Because we know kids eat cereals. Many kids eat cereals every morning for breakfast. Cereals, which come from oats and things like that, tend to have high levels of glyphosate, especially the oat-based cereals. The reason for that is many farms still use glyphosate as a desiccant to dry out the crops before harvesting, right? It makes it easier to harvest a crop when they're dry, so they actually coat the fields with glyphosate in order to dry out the crops. And so then the oats that are derived from it tend to have very high levels of glyphosate. So then we said, okay, let's take what levels of glyphosate that the EPA designates as safe, right? And expose a healthy prime pediatric microbiome to that over a 3-week period and see what changes, right? So we had 3 weeks of stabilization and control on that 5-year-old's microbiome. And what we did, we didn't feed the 5-year-old this, of course. We used a SHIME system, which is a simulated gastric system, which is an entire mechanical digestive tract that has a mucosa, and we inoculate it with the full microbiome, and we feed it like you would a normal human from the stomach all the way to the distal bowel, right? And we can dive in there and see what happens to the microbiome over time as we feed it different things. So what you do first is you take that child's microbiome through lots and lots of stool samples, and you inoculate the entire system with the child's microbiome. You stabilize the microbiome over a 3-week period by feeding it an ideal macromolecule-based diet, meaning ideal amount of protein, fiber, fats, and so on. So you stabilize the microbiome, and this child's microbiome was super diverse, very healthy, high levels of keystone species, high levels of short-chain fatty acids being produced, and it looked pristine, right? This child was vaginal born, was nursed over a year, naturally nursed, had never had a course of antibiotics, hadn't had a vaccine just yet, so it had a pristine microbiome. So we take this microbiome, we stabilize it, and then for the next 3 weeks, we feed it the normal stable food that maintained that healthy microbiome, but we added serial level of glyphosate to it. Now, we did 2 studies. We did glyphosate, the active ingredient only, and then we did Roundup, which is the commercial formula that glyphosate is found in, right? And we did that for 3 weeks, and then we measured what changes occurred in the microbiome. And then we did the last 3 weeks where we try to rescue the microbiome and recover it despite continuing to expose it to Roundup or glyphosate. Here's what we found. In 3 weeks of glyphosate exposure of just the active ingredient, there was some damage to the microbiome, but not a lot, right? But the Roundup version, the commercial version that has other acids and things added, was devastating to the microbiome in that 3-week period. We saw a significant reduction in diversity. We saw a significant increase in pathobionts. We saw a significant reduction in the production of short-chain fatty acids. We saw keystone species dropping. We started to see a high production of things like ammonia and branch-chain amino acids, which are very inflammatory. So basically, this pristine 5-year-old microbiome, within 3 weeks of exposure, started to look like the microbiome of somebody with inflammatory bowel disease, right? And this is the quote-unquote safe level of exposure. Now, we started trying to rescue it by adding in spores and other things, and we started to see a repairing occurring, right? So then that also tells you what you can do about it. So I would say what you can do about the issue of being exposed to pesticides and herbicides is to choose organic as much as you can. You will still get some exposure, even through organic, but it will be less. And dose and frequency matters, right? So try to choose organic as much as you can. The other thing you could do is also try to grow some of your own produce, like we talked about earlier, right? Simple stuff, potatoes, cucumbers, jalapenos, things like that. So growing some of your own so that you reduce exposure from those types of produce that you would normally buy. The third thing is washing. Now, in some types of vegetables and all, washing the surface will take care of it, right? Vegetables with hard peels on them. So if you look at like a cucumber or carrot that you have to peel, the outer surface, those surfaces tend to do a good job of keeping the glyphosate and those chemicals on the surface. You wash those with water and you're typically okay. Some people suggest soaking many of these vegetables in things like apple cider vinegar. I don't see any harm in doing that. I haven't seen a study that shows that it pulls out the toxins, but I don't have any issue with doing that. We do that with some of our produce as well. So that's something you can do as well. Now, know that inevitably you're still going to get exposure to these herbicides and pesticides, especially the commercial version of Roundup. So you need to constantly be working on repairing your gut microbiome, which means the feeding, the fasting, the going out in the environment, taking the right probiotic. We tested in that study the spore probiotics, and we saw that the spores were able to start recovering the damaged microbiome despite continuing to expose it to the Roundup, right? So that's something you can add in as well to try to minimize damage.
You mentioned before the salmonella being picked up by the tomato and being within the flesh, and you can't even see it. As you're sharing about peeling the vegetables of the pesticides and herbicides, do you have any idea of how much ends up like in the middle in the flesh?
With the vegetables that have a hard outside coating or a pretty firm outside coating, the vast majority. It's hard to say a number. Nobody's done a study on the number, but the vast majority ends up on the outside surface. So if you wash the outside surface or you peel it to a certain degree, I think you're doing better. We peel all our carrots. I would love to be able to eat the outside because, again, the outside part, the very outside layer provides more fiber. But if we're not growing the carrots ourselves, we know that it probably has some degree of pesticides and herbicides on it. So we peel it and then you boil it. That can help as well. There are some vegetables that we do soak in apple cider vinegar, things that are like very leafy things, right? And the problem with leafy things is just there's so much surface area, right? So if you buy like a whole head of cabbage, for example, right, there's so much surface area on there for the not only the pesticides and herbicides to sit, but even microbes, right, that may be harmful to you can sit there. So we do soak those as well.
When we were talking about diversity of food and the importance of that for the microbiome, you did mention meat. And classically, when people talk about eating a diverse rainbow of colors, they're referring to fruits and vegetables to get, that diversity within the microbiome. So let's talk about the animal protein piece and how that fits in and how important that is in the full spectrum of building up the microbiome.
Yeah, to me, protein is an important component in itself, right? In two different ways. So there are microbes that will metabolize proteins themselves and utilize them for energy and growth and producing byproducts and so on. So they do directly feed certain species within the gut microbiome. But then microbes also need amino acids to function, right? Just as our cells need amino acids. So providing those amino acids to the microbial community becomes really important. If your protein intake is really low and you're not providing your microbiome with adequate amino acids, it can't build its protein structures, right? Microbes are constantly making proteins, and in part making proteins for us that we utilize. Enzymes are all proteins, right? So microbes are making enzymes all the time to break down things like carbohydrates. But in order for the microbe to be able to produce the enzyme, which breaks down the carbohydrate, which then allows the microbe to utilize the carbohydrate, that enzyme is made up 100% of protein, right? So the microbe needs access to amino acids. And it can't necessarily synthesize its own amino acids because there are a number of amino acids that can't be synthesized from any other starting material, right? So you can look at protein both those ways. One, as a way of feeding the microbes where they directly utilize the protein. Number two is supporting microbial systems that require amino acids. I always like to get as much diversity in the amino acids as well, right? So there's something called the PDCAS score of amino acids. Mother's milk is thought to be like the most ideal diversity in protein profile in terms of the different types of amino acids that are there. So mother's milk would have a PDCAS score of 1. Right, that's an ideal protein, meaning it has all the essential amino acids and everything else you need. Now, anything that's, let's say it's PDCAS of 0.98, that means it's of 98% similar to mother's milk. There are some proteins that are over 1, which means it's even more diverse in terms of amino acids than mother's milk. And so as I'm looking at my dietary proteins, and I supplement proteins as well because I lift, right? So if I'm lifting, if I'm doing resistance training, it's really great to get a big dose of free amino acids in your system after you lift. Studies show that the moment you're done with the workout routine, if you increase the amount of free amino acids in your system, it creates this myogenic activation, which is that activation of muscle building, right? So for that reason, I supplement protein. But when I eat protein, and I eat protein from animal sources, I try to get a diversity in that too. So lean beef, right? Pork has different amino acids than beef does. Chicken, lean fish. I love fish, and I love a lot of lean fish. I love sushi, for example. So I try to eat a lot of sushi grade fish. I try to get a diversity of the protein from those as well. Now, you also get other compounds that come along with it. You get peptides, you get nutrients, micronutrients like vitamins and certain minerals that come within that protein as well. And at the end of the day, one of the things that we have to accept, because diet becomes very polarizing, right? And it's one of those very difficult topics to talk about, because for some reason people become very activist like about their diet choices, right? Whether they are paleo or keto or vegan or vegetarian, whatever it is they choose, whatever camp they're in, they're very activist like about their position on it. The fact of the matter is humans are omnivores, right? All the evidence we have in terms of our physiology, our guts, our microbiomes, looking at other species that are similar to us in the environment, they're all omnivores. They're roaming omnivores, right? Just a couple of pieces of evidence for that. So if you just look at our dental structures, we have both incisors and we have masticating teeth, and we have canines that can cut meat, right? Very few mammals have both, right? Unless they eat both meat and they eat vegetation, where they need a grinding capability and they need cutting capability as well. If you look at our digestive tract, so when the herbivores, obligate herbivores, tend to have multiple stomachs, right? Because they have 4 or 5 different stages of fermentation to be able to utilize all of those plant-based materials fully and effectively. We don't utilize a lot of the components within plants very well. And so we have what we call a medium length gut with a big fermentation at the end, right? Our small intestines is more predominant than our large bowel in terms of length and size. And that's because fermentation is secondary to assimilation of nutrients. And that's because we can assimilate nutrients from lots of different sources, right? If you look at carnivores, obligate carnivores, they have much shorter guts and they don't have as much fermentation in their system, right? So there's lots of evidence. And you look at the animal that actually has the most similar microbiome to a human is a baboon, actually. So we are genetically closest to chimpanzees, right? We're like 98%, almost 99% similar in genetics to chimpanzee. And yet our microbiome is actually quite a bit different than chimpanzees. Our microbiomes are much closer to baboons. The reason for that is baboons are much more omnivorous than chimpanzees are, right? Baboons hunt, they eat meat, they pick, they forage, they eat insects, they dig, they eat roots and tubers. They also roam, right? They move in tribes, in groups, and they roam throughout environments similar to what humans did. And that's why their microbiomes have evolved similar to ours.
Given what you just said about humans being omnivores, there is this growing trend of people in the health world only consuming meat, or you mentioned carnivore there, going on a carnivore diet. And seemingly, they've been doing really well when it comes to healing from chronic disease. Have you looked specifically at the microbiome piece, somebody that's been on that type of diet over a long period of time, and what happens? And you mentioned before the fasting piece. We could maybe tie that in at the end, because that would be a pivot where we're actually giving no food to the microbiome. In this case, we'll start with just meat.
Yeah. So there are some studies coming out that show that if you go full keto, for example, or full carnivore, it starts to really impact the diversity of the microbiome, and it doesn't take a lot of time. It can be 3 months to 6 months where you start to see a reduction in the diversity of the microbiome, which means if you look at all of the studies on the importance of diversity and longevity, so you can actually predict someone's longevity based on the diversity of the microbiome at a given age, right? It's one of the most accurate and best predictors of longevity and disease resilience. So that kind of diet, reducing diversity, even though you may see certain short-term improvements, and we'll talk about why you might see short-term improvements, but because it reduces diversity, to me, that's a long-term misstep, right? I think you will pay for it down the road. Now, why is it that people go carnivore and then they start to lose weight? They feel better, the metabolism, they have less inflammation, and so on. Well, part of that is because on a carnivore diet, there are a couple of things that'll happen, right? Number one, you're basically eliminating sugar from your diet, right? And it's easy to eliminate sugar when you go on a carnivore diet because you don't have any options that actually have sugar in it, right? Unless, of course, you're mistakenly eating like, fast food meat that is marinated in sugar-based marinades and all that. But that's not what the people are doing, right? When they eat carnivore, they're roasting chicken, they're eating turkey, they're just eating steak for the most part, and you're just grilling or cooking steak, smoking steak, and eating it with a little bit of salt on it, right? That becomes an easy way to eliminate sugar. That's one of the reasons why people's systems start to get better. They start to lose weight. You also reduce caloric intake, right? Because a carnivore diet is very high in protein. Protein provides a lot of satiety, right? And so you don't actually overeat. So you end up with caloric restriction and almost no sugar in your diet. You're going to lose weight, right? And when you lose weight, inflammation comes down, right? So that's all a beneficial thing. And maybe you can do it for 3 weeks. Maybe you can do it for a month, right? And just to get your metabolism and kick started in some way and kind of get yourself feeling better. Let's say it's early spring now, and you're like, oh, I want to get into bathing suit shape, right? Maybe you can kickstart that with going carnivore for a little bit. And that might give you enough effect that keeps you motivated, right? But if you do that forever, if you do that for 6 months, 8 months, 10 months, 12 months, you're going to start reducing the diversity of your microbiome, and that will have health implications down the road, right?
How do you feel about using that as a kickstart if somebody's coming to this conversation, eating a standard type diet and their microbiome's a total mess? You mentioned it more in a general sense being a kickstart, but can you see that being a good way to balance out the microbiome before taking on a lot of the principles we're talking about today?
I think it could be. I think it could be, especially if someone's diet was a mess before. They've had lots of antibiotic courses. And let's say they have low diversity already, right? Their pathobiomes tend to be high, which means they're probably sensitive to a lot of foods anyway, right? So a lot of plant-based foods may actually cause them distress because their systems aren't capable of handling it. So in that case, you could eliminate a lot of those things, go carnivore for a short period of time. I would say 3-4 weeks, right? To kind of start to reset your system. Your diversity is not going to improve in any way at that point, but your system, you might feel better and your system might start to function a little bit better. But then you want to start adding in other foods relatively quickly, but at small amounts. Right. So I would go, if I was doing this, I would go, okay, I'm going to go carnivore for 3 weeks. At the end of that 3 weeks, I'm going to start adding in some nuts, some seeds. I'm going to start adding in some polyphenols. So a serving of fruit with my meal, right? I might eat a steak still, but I might have steak with blueberries to finish it off, right? So start bringing in the diversity of food slowly. Because keep in mind that our bodies can adapt, our microbiomes can adapt to almost any diet that you put it through, and it'll find a way to function. It's not going to function well, but it'll find a way to give you basic functionality. But if you start adding in all the right foods at slow amounts, at a certain pace, you'll start to diversify your microbiome and start to build that resilience. So I think it could be, I would just, if it makes people feel better, makes them lose a little bit of weight, get more energy and so on, I think it's fine. But long term, it's not something I would do. I just heard, Joe Rogan on a podcast, he was talking about when he went carnivore. He did it for about a month, right? And he said he lost weight, he became leaner, but the problem he was having an energy issue. When he did hard workouts or longer workouts, he was wiped out. He had no energy, right? Of course, because you need carbohydrates for energy. You need to build your glycogen storage. You need to build muscle glycogen, liver glycogen. That's what's going to give you a lot of energy throughout the day. Your brain uses a lot of carbohydrates. Sure, you can end up going ketonic and start making more ketones, but your brain is actually naturally designed to use a lot of carbohydrates for normal respiration and ATP production. So you don't want to necessarily do that for long term, but maybe it's a kickstart.
All right. So when it comes to the diet, we want diversity, we want plants, we want meats. How do you feel about fermented foods? Things like live sauerkrauts, yogurts, kimchi. we could go on and on. There's a lot of different ones available these days. The general assumption with those is that these bacteria that are still alive within those foods are making it down to our digestion and repopulating. So two different parts there. Are they beneficial in a general sense? And then two, do they actually repopulate like people assume?
So most people will consider fermented foods as a probiotic food, right? And I was invited to do a talk at like the Santa Barbara Fermentation Festival, big fermentation festival. They're like, oh, here's a microbiologist with all this microbiome knowledge. We'd love him to talk. My first sentence in my talk, and I thought either A, I'll be chased out of there with pitchforks, or people will listen. My first statement was fermented foods are not probiotic foods. They're not a source of probiotics, right? And people were like, what? What the hell is he talking about? We thought that was the whole benefit from it. Well, it's not. The point of fermented foods is not to deliver bacteria to your system, because those bacteria are designed to ferment the food outside of the body. And they do so under their own specialized conditions. They don't function inside the body. The moment you eat them, they're all going to die in the gastric system. The stomach acid's going to kill all those bacteria. So it's not about delivering bacteria. What it is about, it's about delivering super nutrients, right? So fermented foods are foods that now contain nutrients that the food naturally did not contain. And those new nutrients come about from the fermentation process, right? You're putting bacteria to break down the normal substrates of the food, and they're creating byproducts that are now super nutrients, right? These are similar to the byproducts that will be created in your gut, right? Remember, your large bowel is a fermentation vessel, right? So let's say you take sauerkraut, right? And a fermented sauerkraut. Let's say you had a non-fermented version of the substrate itself, the vegetable. You eat that. What's supposed to happen is that's supposed to go into your large bowel, and for the next 8 to 10 hours, it gets fermented and becomes sauerkraut in your large bowel, right? Now, assuming you don't have enough diversity, the conditions aren't right, you may not get adequate fermentation out of it. But doing it outside of the body, in a way, and then consuming it gives your system an edge, because you are producing all of those wonderful nutrients that come as a result of the fermentation, right? So what are the things that are produced during fermentation? Well, microbes that ferment certain foods will take the basic substrates like the carbohydrates, the proteins, and the fats, and they'll produce things like peptides. Like dipolonic acid, for example, is a wonderful peptide that can reduce inflammation and help circulation and help blood pressure and all kinds of things. They produce urolithins, which help your mitochondria repair themselves and kick off autophagy and mitophagy. So removing damaged cells and cellular debris. They produce short-chain fatty acids like butyrate, right? So they produce all of these, and an array of vitamins and minerals and all of these things. So fermented foods are basically nutrient-dense packed foods that can be very beneficial, assuming that you're not sensitive to that particular type of ferment, right? Because some ferments will have high levels of histamine in it, which is a natural byproduct of the fermentation. So some people with histamine intolerance, for example, may consume that, and they may be reactive to them, right? So if you have a fermented food that you can make yourself or somebody around you makes, or that you can buy from a somewhat traditional source, and you consume it and it agrees with your system, it's perfectly great for your system, right? I would always encourage people to consume that. There's certain fermented foods that really work well. Like I love kimchi, right? I would put kimchi on anything. I come from South India, and in South India, we have a lot of fermented lentils. And in fact, we make breads out of the fermented lentils and so on. So that to me is very staple for my system. But some people may not, sauerkraut or kefir, things like that may not agree with them. If it doesn't agree with you, don't do it, right? Find a fermented food that does agree with you. And if you can create it yourself, that's really the best, right? When you look at an example of that is kombucha, natural, actual made kombucha using traditional techniques can be very beneficial for you, right? Because they have all these amazing organic acids in them. And organic acids are really good for the gut. But store-bought kombucha in the bottle is not even kombucha, right? They put the label of kombucha on it, but they don't complete the fermentation because the fermentation takes too long. It makes it less commercially viable. They add a bunch of sugar and flavoring and colorants and things like that in it. So then it negates a lot of the benefit, right? But if you can make your own kombucha at home, do that.
As you're talking about these different nutrients that get unlocked when we ferment, and then we still get benefits from taking those in, but again, not the benefits that a lot of people would assume from the bacteria being alive. It gets me thinking about the difference between creating those nutrients within the colon versus taking them in, having to go through the stomach acid, small intestine, and then get to the colon. Things like, the peptides you mentioned and different nutrients, are they stable to go through that and to get the benefits when they get down to the colon?
When you think about the nutrients that come in from the fermented food, so the peptides, the enzymes, the fatty acids, the organic acids, all of these beneficial things that are being made during the fermentation process that we want as nutrients, whether those make it through the gastric system or not, the answer is pretty clear that they actually do, right? So the gastric system doesn't dismantle nutrition so much. The gastric system has two real big functionalities. Number one, it's to kill off organisms so that we don't unnecessarily bring in huge loads of viable organisms. And I think more so from food, what we're trying to protect against are microbes from the mouth. Right. And I'll elaborate on that in a second. But we want the gastric system to act as a gastric barrier to kill microbes that are trying to come in through the digestive system. And then the second part of it is to aid in digestion, especially in the digestion of protein. Right. So the gastric system really, really is important for the digestion of protein. So if you think about a piece of chicken, for example, the protein in the chicken is a string of amino acids, like think of a string of pearls, but it's folded in all of these complicated structures, right? It's called secondary and tertiary folding. So the amino acids and the protein is not actually accessible to us in that structure, right? It's incredibly hard to get to in that folded structure. So what needs to happen is that folded structure needs to be unraveled, and the stomach acid helps do that. So there are all these bonds that hold a structure folded. They're called van der Waals forces and hydrogen bonds. And then acid breaks up those bonds. So they go from a folded structure like this to a long string of pearls of amino acids. So then from your stomach, the protein moves into the small intestines as a long string of amino acids, right? And then enzymes go to work on it. You've got peptidases and proteases that start chopping up that long string of amino acids into individual amino acids, which is what we assimilate and absorb. Now, the microbial part of it, the microbes on the fermented foods and the microbes from raw foods, like if we eat, something off our garden, even though those microbes are getting killed in the stomach by the stomach acid, which is what we want, you actually still get some benefit from those microbes because of a couple components. Number one, the internal cell of the microbe. So you think about the microbial cell like this, right? It's a whole cell with a defined structure. What the acid does is it actually causes a cell to blow up, right? So it's called lysing the cell, which is blowing it up open. So now the internal components of the cell are now exposed to us, and there's lots of beneficial things in that internal component. There are RNA components that can affect gene expression within us. There are enzymes, there are peptides, there are lots of things that we can utilize inside that cell. We may absorb and utilize it in our own needs. The other thing is the components of the cell. So the outer components of the cell that's now in pieces, as that moves through the digestive system, it gets sampled by the immune system. That's how the immune system learns what kind of microbes are in your environment, right? And it builds tolerance against those microbes if it's microbes coming in with food, right? It goes, this is a normal microbe. It's not a pathogen. I don't have to go crazy and attack it every time I see it. I'm going to build some tolerance against it. So I allow it to come through, but I monitor it, right? And this is how your system adapts to the environment that you're in. Now, I mentioned the oral microbes. The oral microbiome is full of problematic organisms, right? They're fine when they're in the oral cavity, but if your gums become leaky and they leak into your circulation, that becomes a big problem. Or if they're allowed to move through the digestive system in a viable state, they can cause a lot of issues. For example, if you look at SIBO, SIBO is a very prevalent condition, right? Small intestinal bacterial overgrowth. There's a fundamental shift in the types of microbes that predominate the small bowel in people with SIBO versus people who don't have SIBO. When you look at what those microbes are, there's a lot of Enterobacter, Enterococcus. These are not normal microbes that are found in the small intestine, but these are microbes that are found at high levels in the mouth. And so people with SIBO often have issues with their gastric system. They're producing low levels of stomach acid. So many of these microbes that you swallow every day through the gallons of spit that you swallow, those microbes are not getting killed in the stomach. They're making it to the small intestine in a viable state where they can then proliferate and cause small intestinal bacterial overgrowth. This is why there's something called PPI-induced SIBO. PPIs are proton pump inhibitors, right? Proton pump inhibitors reduce stomach acid production. It's used as a way of dealing with symptoms for people that get reflux, right? And so when you reduce stomach acid, you end up with SIBO because these microbes are going in a viable state.
So when it comes to SIBO, I would assume if somebody's not on one of those drugs, part of fixing that would be to rebuild up stomach acid in a natural way.
Yep.
So let's talk about that and how we do that. And then also other players in the SIBO realm and what we can do to correct that if we have that condition.
Yeah. So two things you want to really address if you have SIBO. One is stomach acid, and the things that really suppress stomach acid production. Of course, the medications we talked about, but let's say you're not taking the OTC PPIs, right? So these are the over-the-counter acid reducers. If you're not using that, if you're not using things like Pepto-Bismol all the time, or you're not using acid reducers or neutralizers all the time, which a lot of people do, and I think that's probably one of the most prevalent reasons for reduction in stomach acid. But let's say you're not using those. The other reasons for a reduction in stomach acid production are stress. Stress is actually one of the biggest reducers of stomach acid production. Micronutrient deficiency, so magnesium is really important for inducing the production of hydrochloric acid, and also H. pylori infection. Helicobacter pylori infection in the stomach is another way of reducing stomach acid production. So you may want to address Helicobacter pylori issues, right? And so that's one aspect of it. You need to start thinking about stomach acid. I would even say if you've had SIBO for a long time, you have bowel movement issues, you get a lot of bloating and all that, you want to consider even using betaine HCl, which is a supplemental form of hydrochloric acid, right? I would use that with my food in order to help digest the food properly and also provide some hydrochloric acid protection during the digestion process. Now, the stomach acid is one thing with SIBO. The second thing is bile. Bile salts produced by the liver are really important in maintaining low levels of microbial growth in the small intestine, especially during digestion, right? So a lot of people with SIBO, a very high percentage, about 4 or 5 times higher than non-SIBO people, also have liver dysfunction. Right. So if you have SIBO, you want to talk to your doctor about getting some liver function testing done and then supporting the liver. Some of the basic things you can do to support the liver are things like milk thistle. Right. Milk thistle, which is also known as silymarin, is really effective at supporting the liver. You also want to manage leaky gut. Most people with SIBO tend to have leaky gut. There's tons of videos on how you do that. We use spore probiotics, prebiotics, a combination of that to manage leaky gut, because leaky gut drives heavy dysfunction in the liver. And if you're not producing enough bile acids, you may also supplement with that. There's a product called ox bile, which is a bile acid supplement, which you can utilize that can take the place of the lack of production of your own bile acids until you get your liver functioning properly.
Have you heard of Dr. William Davis's SIBO yogurt? And
I have.
If you have you tried it?
I have. I actually have. He sent me some of his reuteri. So he uses a Lactobacillus reuteri, which is the key organism in the yogurt. He also has a probiotic with the high amount of that ruteri. And he also provided me once instructions on how to use that probiotic to make the yogurt. I think it's a fantastic product. I think it's great for the system. I think it can help with SIBO because it can compete against some of those overgrown organisms. And I think part of it is also it is soothing to the bowel, so it can help alleviate some of that leakiness in the gut and get the bowel functioning properly as well. But yeah, that's a great approach to it. And ruteri is one of those really important keystone species that is almost eliminated from the Western population. We have almost no ruteri in our system at all. And it's very high in prevalence when you look at hunter-gatherer tribes like Papua New Guinea tribes and the Hadza tribe and so on. So that's one of those organisms I think that has suffered through, industrialization and overuse of antibiotics and all that, and we've knocked it out of our system to a certain degree.
You mentioned the spore-based probiotic, and we're going to get into that. I know you're a fan. Let's talk about somebody going to, say, a grocery store or health food store, going to the refrigerated section, getting a classic probiotic capsule or powder, and talk about what happens when we ingest that, and then we'll move into the spore-based.
Yeah, and here's an important thing for people to understand. Why is it in the refrigerator to begin with, right? How did they figure out they want to put this product in the refrigerator? Well, as it turned out, when people started, producing and releasing probiotics, this was really starting to pick up steam in the late 80s and early 90s, and then really grew in the late 90s and early 2000s. The term probiotic was really coined in the 1960s, but probiotics weren't really a component of the health food industry until that late 90s, early 2000s period. Now, here's what happened. People started taking fermenting organisms, right? So take Lactobacillus acidophilus, for example. It's the most common probiotic strain out there. Why is it? Is it because it has some special function inside the body? No, it's because it was the most common fermentation strain, right? Acidophilus, as it suggests, is a microbe that likes that functions better at lower pH. It likes acid more than other bacteria, and so you can ferment it at a lower pH. You can ferment dairy, for example, at a lower pH with acidophilus. That actually produces a better tasting dairy ferment, less sour in general, right? So acidophilus became synonymous with fermentation. And then fermentation was mistakenly thought of as a probiotic source. So then somebody said, well, I'm just going to take the acidophilus that we use in fermentation, put it in a capsule, and make that a probiotic, right? And so that's how some of the initial probiotics got started. They were fermentive bacteria that are used to make fermented foods that now were isolated and put into capsules. The problem is these microbes don't function in the intestines. They function outside of the intestines in a fermentation factory. And so taking them on their own doesn't really have any benefit. But then, of course, in our megalomaniacal society, people said, well, if, 1 billion of this fermented bacteria is good, maybe 2 billion's better, maybe 10 billion's better. Then all these companies started competing with each other on this billions of CFUs idea, right? They started creating this false notion that the potency and efficacy of a probiotic is dependent on its CFU count, when it actually is not, right? So now we started getting 50 billion, 80 billion, 100 billion CFU products. They were just competing with each other more and with higher and higher doses. However, what started to happen is most of these organisms are very unstable. They're not designed to be on a shelf. They're not designed to go through the gastric system. And so when they started fermenting or formulating these products, putting say 50 billion in a capsule and putting it on the shelf, those microbes started having high attrition rates, meaning they're dying in the capsule because they're such unstable organisms for that type of system. And so then class action lawyers started saying, hey, this company is saying there's 50 billion colony forming units. That's what CFU is, right? CFU is basically you take the bacteria, you plate it, every single cell will form a single colony. So if you have 50 billion colony forming units, you should be able to plate it and count that on a plate. And so what the lawyers are doing is grabbing these products off the shelf and testing them to see if they actually contain 50 billion CFUs. Well, most of those products would face massive amounts of attrition, so they might now have 10 billion CFUs, 5 billion CFUs, and then these companies are getting sued for false advertising, right? So then the company said, oh, okay, well, what should we do in order to preserve these microbes so they don't die? Well, one of the ways in which you preserve microbes and stop them from growing and dying is by refrigerating them, right? So that's where the idea of refrigerating these probiotics came from. So they did two things. They said, we're going to put overage. If we say 50 billion per capsule, we're actually going to put in 200 billion so that we have some slack there. And then we're going to refrigerate it so that the microbes are suspended and stuck metabolically and they won't die. So microbes, when you refrigerate them, don't multiply and they don't die either, right? They can stay in the suspended animation. And so that was a whole reason for this refrigerated probiotic nonsense, right? It has nothing to do with efficacy. It has nothing to do with science. It has nothing to do with research. And then all this high dose was all marketing as well. The vast majority of probiotic studies are normally done with a single strain at 3 to 5 billion CFUs, and they're not refrigerated, right? So that was just a very misleading approach to how you think about probiotics, right? It's not the number of species. That doesn't matter. I would rather have one viable functional species in the probiotic than 15 nonsense species in there that the company has no idea how they work together, whether they knock out each other's function, they compete, or they're all just dying. And I would rather have the clinically efficacious dose of 3 billion, 4 billion, or whatever the studies have shown versus just having something with hundreds or 50 billion in there, right? So if you go and you're told that the refrigerated probiotics are the best and the highest quality, you take those home. As soon as you pull them out of the refrigerator, they'll start to die. While you're driving in your car to your house, they're dying. While you put them in your cabinet, they're dying. And every single day that they're sitting in your cabinet, they're dying. And then the moment you take them and you ingest them, any of them that are still alive in the capsule will be obliterated in the stomach acid. And so you're getting a bunch of bacterial debris, not really a probiotic, because there's a scientifically accepted definition of the probiotic that was put forth by the WHO that's widely accepted by all researchers in this field. And that is a live microorganism when administered in adequate amounts, confers a health benefit to the host. So that first part is really important. It has to be a live microorganism.
So it sounds like it would be a lot like the fermented food, where there would be certain benefits from the bacteria, but we can't expect it to get to the colon alive.
That's right. Now, in the case of what I call kitchen sink probiotics, there are a couple of dangers with them as well, right? So normally in fermentation, you don't have 15 different strains represented in the product, right? Because fermentation is normally done by one species. And so if you have a fermented dairy, you've got one species that's doing that. So when you consume it, you're consuming the one species and all the debris of the one species. The thing with these kitchen sink formulas with 15, 16 different species in there, the problem that exists is many of these species can be inflammatory. And in fact, we've checked this. When one of my last projects, when I was at Microbiome Labs, we were working with University College Cork and researchers at the APC, and what they were showing is many of the commonly used strains as probiotics actually increase inflammation. They trigger a whole cascade of inflammatory responses, right? So when the debris is coming in at that high dose, it can actually make you more inflamed. In fact, when you talk to clinicians, many clinicians didn't use any probiotics with patients that have really inflammatory conditions, right? Think about patients with mast cell activation syndrome or histamine intolerance or SIBO even, right? Clinicians weren't using probiotics with them. Why? Because in most cases, these probiotics made them feel worse because these probiotics are actually activating a whole series of inflammatory responses in the body. Now, we also know from studies from the, I think the university, there's a international university in Israel that showed that these kitchen sink type formulas can actually compete for binding sites with your own natural versions of those species. So taking them, for example, after the course of antibiotics will actually slow down the recovery of your microbiome after the course of antibiotics, right? So in my view, there's almost no need for a hodgepodge kitchen sink probiotic with 15, 18 different species in it, because there's no good scientific rationale behind them, right? 4 or 5 species, great, especially if you demonstrate in clinical trials on the finished formula that these 4 or 5 species work well together and they have a additive effect in the system, right? Many other good probiotics will be a single species in them. Think about Saccharomyces boulardii, right? Florastor has been around forever. It's got something like 75, 80 published studies on it, and it's a single organism. It's a yeast probiotic, but it's still a single organism, right? Think about the most common from Procter and Gamble, which is Bifidolongum 35624, which is one of the most well-studied organisms for IBS, right? And longum is a keystone species that has almost 80 published studies on this one strain. That's the number one selling probiotic in the U.S. market and it's a single strain, right, at 3 or 4 billion CFUs, because it matches all of the clinically relevant data. So this whole idea of just throwing stuff together and saying, hey, we have 15 strains and 50 billion CFUs, it's all marketing nonsense.
So when it comes to longum or Florastor, it sounds like you're a fan of those. Are those refrigerated ones?
They're not.
Are they just, okay. I just want to understand the nuance there. Is there anything in the fridge you would take?
No, I don't see any benefit to any of them that need to be refrigerated, right? To me, if they need to be refrigerated, the assumption is the viable cell is important for functionality. But that cell is not going to be viable the moment it hits my stomach anyway, right? The longum is an interesting one, right? So P&G, the company that has the longum product out there, and we own that longum strain, actually, that 35624 strain. When I say we, I mean Microbiome Labs and Novozymes, not me personally, but that strain is very, very interesting because we took care to try to preserve it and manufacture the product under low moisture environment, low shear, and so on, so that we would preserve more of the organism, right? So that you're sending in viable strains into the system. However, when you heat kill that organism, you find that it has very similar efficacy, even though it's dead. And certain organisms do that. Now, the reason is because that particular organism, the thing that provides it its function, is a very unique carbohydrate that the organism produces that's on the outer cell wall of the outer cell membrane of the bacteria, right? So the bacteria produces something called peptidoglycan. Peptidoglycan is a very specialized carbohydrate structure that's embedded in the cell wall of the bacteria, and they have these what we call them polysaccharides, are just meaning many different carbohydrates that are sitting on the outer surface of the microbe. Now, those polysaccharides and peptidoglycans were what conferred all of the health benefits of that organism. We know that because we did studies where we took the organism and we knocked out the gene that makes those polysaccharides, and then we tried them in animal and human studies, and they had no effect, right? And then we isolated the polysaccharide and tested that, and that had all the effect. Right. So that kind of organism, it doesn't matter actually if it's alive, because a polysaccharide is on the outside of the organism. If it goes in and the stomach acid obliterates the organism, it releases the polysaccharide, actually. Right. And the way the organism actually functions is actually really interesting. So imagine it's a viable version of the organism. The cell is intact. The polysaccharides are all on the outside. It makes its way down the small intestine. It gets to the end of the small bowel in an area called the Peyer's patches. In that area, you have dendritic cells, which are some of your immune cells that reach across the lining of the gut and grab things, right? And sample things that are there. When it recognizes the presence of this organism, it actually grabs that organism, pulls it across the lining of the gut, and brings it into circulation, right? Now, Normally, a dendritic cell is what we call an antigen presenting cell, because what it does is it eats things that are potentially harmful, digests the things on the inside, and then presents some component of that cell to your T cells and B cells so they can mount a response. So that's how dendritic cells function. But when the dendritic cell consumes this microbe, it doesn't present anything to the T cells and B cells. It doesn't activate the immune system. Instead, what it does is it digests the microbe and spits out all those carbohydrates into circulation. That's how the carbohydrates end up in the neurological system and reduce inflammation and all that, right? So you need this interesting nuance and elegance of certain strains that allow them to work under many different conditions. You can't just throw together a bunch of random species and assume it's a probiotic.
All right, let's move into spore-based probiotics and why you're a fan of these.
So, what I liked about spore-based, and we pioneered spore-based probiotics, right? So what's interesting is prior to us launching them, this category of spore-based probiotic didn't exist. We created the category essentially. And to give you the short version of the story of how we came about this, the idea was, I'm always looking at evolutionary biology for cues as to what we should be doing from a supplement nutritional perspective, right? So when it comes to bacteria, that's even more important, because I was trying to look at, okay, what is our natural relationship with microbes, right? So when we are in utero, we get some inoculum from mom's gut bacteria. In fact, dendritic cells reach across, pull certain bacteria from mom's gut, and take it to the baby in the amniotic fluid and through the cord blood. So you get some inoculum. Then when you're born, you get a huge inoculum passing through the birth canal. You get inoculums from skin-to-skin contact, and you get inoculums from nursing. All right, nursing provides lots of different microbes. So that's all within the first year-ish of being born. That's the majority of microbial exposure. But then the next subsequent years and the rest of your life, your exposure is through the environment, right? And if you think about an interesting feature of babies, and this is something that I've always thought about and a lot of thought experiments on, is babies have a tendency to put everything in their mouth. Right? They sample their environment with their mouth. They try to figure out the world around them with their mouth. And that's a weird behavior, because as a species, our mouth is not a sensory organ for us, right? We are tactile and visual, and we might smell things, we might listen to things, right? If we're trying to figure out what's in a gift, for example, we might shake it and listen to it. We might smell it, right? We look at the shape. We don't lick it to try to figure out what's in the box, right? And so when you think about, well, why do babies have this natural tendency to put everything in their mouth? Well, it's because they're inoculating their system with the environment, which is so important for the development of their microbiome. So if you think about an ancestral baby is born, is put in the dirt, it's putting rocks and dried dung and dirt into its mouth, right? So that's a diversification of the microbiome early on. So then it becomes really clear that what's very important for the function of our microbiome is engagement with the outside environment. Now, when you look at most microbes in the outside environment, most of them don't function as a true probiotic, because most of them will get killed in the gastric system, right? And you're getting the debris, and the debris is engaging with the immune system and so on. But what if there were microbes that actually have a natural capability of surviving through the gastric system, and they're in the outside environment? Are they the true probiotic, right? And so that's what we went in search of. And that's why the spores come in. The spores are so interesting because they're ubiquitous in the environment. They can exist in the environment in that spore state, which is a suspended state. They are not metabolically active. They're not multiplying. They can sit in the dirt for millions of years, literally millions of years. Then you consume them, and that spore state allows them to survive through the gastric system. But the moment they hit your small intestine, they come out of the spore state, and they start functioning as a functioning probiotic in your system, right? And they're designed by nature to be able to do this. There's very, very few microbes that can survive the gauntlet of the gastric system, right? Because it's designed so specifically to kill microbes. And here is a natural microbe that nature has given the ability to survive through the gastric system. And our system, our immune system in our gut recognizes them as friend, because they don't trigger inflammatory responses in the gut as well, right? So those are very important features. And we said, okay, these must be what nature intended as probiotics. So let's study them a little bit more. Let's study the impact they have on the rest of the microbiome. Let's study the impact they have on the immune system, the tight junctions, the leakiness in the gut, all of that. And we ended up publishing about 18 studies on the spores, showing that they have a very, very huge impact on the health of the microbiome.
And when it comes to the spores, we're talking about supplement form here in general. How, in a general sense, would we engage with them if we weren't taking a supplement?
Yeah.
Like, do they come in on different types of food, being in different environments? Basically, up till this point of having access to these supplements, how much engagement did humans have with spores day to day?
Yeah, we had a ton. So spores in the outside environment, you take almost any sampling of natural soil, you'll see spores at the rate of around 10 to the 4, 10 to the 5 CFUs per gram, right? What that means is 10 to the 4 is around 1,000 colony forming units per gram. 10 to the 5 is about 10,000, right? So if you go out and you take 1 gram of dirt, which is not a lot, it's a tiny amount, you've got about 4 to 5,000 live active spores in there. So there's quite a bit in the outside environment, right? In rivers and streams, dirt on vegetation. If you pick fruits from the tree naturally and eat them, they'll have spores on them. So we evolved in an environment with these organisms, and we created this amazing symbiogenic relationship. So there's a process called symbiogenesis, which is where when two organisms are forced to exist in the same environment, eventually what happens is they develop a symbiotic or mutualistic relationship, because it's better for the two organisms to work together than work against each other, right? So because spores are ubiquitous in the environment, we have this symbiogenesis where we ended up providing them a home. Our immune system recognizes them as self, and so it doesn't attack it. Our system recognizes them as self, so we provide them a home that's not hostile to them. And in return, they clean up the home for us, right? So they do this amazing thing of fixing the gut microbiome. They use something called quorum sensing. Spores have this ability to go into a microbial environment and then read the rest of the chemical signatures of the microbes and figure out who's there and in what amounts. And then they can do specific things to bring down the growth of pathogenic and dysfunctional organisms, and they will do things to increase the growth of beneficial bacteria. We did a study that we published, I think in 2021 or 22, where we showed adding spores to a gut microbiome in 3 weeks increased the diversity of the gut microbiome by almost 25 to 30%. And it increased the growth of keystone species, which is critical, and it brought down pathogens, right? So the spores are like the police or the orchestrator of the microbiome. They do it better than we can do it. So that is the nature of the mutualistic relationship we have with them. We ate them all the time inadvertently. We gave them a home. We said, hey, you're welcome here. We're not going to attack you. We're not going to be hostile to you when you enter the system. And in return, you need to make sure you fix and manage the system for us. And they're temporary. They're transient. They'll stay there for a couple of weeks, and then they defecate out and they go back into the environment. And that's how they find a new host.
You mentioned how common they are in the environment. So it gets me thinking about the advantage of supplementing. Is it that it's so concentrated? Is it that there are different types within the supplement than we'd encounter day in, day out in the dirt on produce? Talk about why that's important to supplement.
Yeah, so the concentration is important, right? It becomes really hard to ensure you're getting the right clinical dose of the spores if you are trying to go out there and methodically expose yourself to nature, right? It's different if you lived in nature, right? If you lived on the side of a mountain in a hut and all that, I would say you don't need any spore supplementation, because 24/7 you're in the environment, right? So you're getting enough dosing. But if you're trying to be prescriptive about it and you go, okay, I'm going to go out and go for a hike and walk 2 or 3 times a week, we don't know if you're actually getting enough exposure to be clinically beneficial, right? We know it's going to be generally beneficial to you, but if you have a gut issue. Is that going to be therapeutic enough? Right? It's beneficial for your gut, but not necessarily therapeutic. So in that case, supplementing with a defined clinically relevant amount becomes really useful. The other thing is the versions, right? So there's lots of different kinds of bacillus spores you'll find in the environment, and not all of them are perfectly suited to function as probiotics in the gut. In fact, the spores that we worked with and we developed, we isolated them from human stool, right? That's where they came from, because we wanted to find versions of the spores that were living in the gut for periods of time. So we know that they're better adapted for the gut environment. Even then, through them, we screened thousands of these strains to find the ones that had the most viability and the most functionality in the gut. So when you take it as supplement form, like in the product Megaspore, what you're getting are the perfectly dialed in strains that have all of the spore features that are important, but are also very well evolved to exist in the human gut, right? And because some of these spores will live in insect guts, will live in worm guts, will live in other things. And so you may not have one that is as functional or viable in the human gut. And what you get in the supplement form is that perfected.
For somebody taking Megaspore, eating the diet we recommended, living the lifestyle you talked about before, is there any other adjuncts? You mentioned again Florastor and another, I forget the other one, but another probiotic. What I'm getting at is if we apply the Megaspore plus the diet plus the lifestyle, anything else as adjuncts we want to put in supplement wise, probiotic wise, to fully balance that out, or does Megaspore have everything we need?
So Megaspore will have most of what you need. I would look at strains that are specific to specific conditions beyond that, right? So for example, the strain we talked about, the Biflongum 35624. If you have IBS and IBS-like symptoms, I would add that in, because that strain is very powerful at controlling IBS-like symptoms and neural inflammation in the enteric nervous system. If you have anxiety, if you're someone that has a lot of stress and anxiety, I would add in Biflongum 1714, which is a psychobiotic that's very powerful at reducing anxiety and reducing stress. We know that stress and anxiety can have a dramatic negative impact on the gut, right? So I would add that to the routine with the Megaspore and so on. I would also add polyphenol supplementation, because you can get a good amount from your diet, but if you're really trying to ramp up your Akkermansia, your keystone species, heal that leaky gut, I would supplement with polyphenols for a period of time. Florastor, I would add that in if you have SIBO. I would add in if you have fungal overgrowth. If you suspect that you have candida, for example, I would add in the Florastor along with the spores, right? So it's specific to specific conditions. So for example, if you're a woman and you have a lot of vaginal dysbiosis, right? You get a lot of BV, you get a lot of yeast infections. I would add, there are certain lactobacillus strains like Lactobacillus crispactus or Rhamnosus GR1 that have studies that show that oral intake of these microbes have a profound effect on the vaginal microbiome, right? So you can absolutely add those in for those specific conditions. And we're just talking about general gut health to maintain a healthy gut, diversify, keep leaky gut at bay. The spores and those other things that we talked about in combination are very powerful and powerful enough.
All right, last question for you. We've talked about the Megaspore. I've worked with Just Thrive before, and I know you've worked with them in the past or are still working with them. You can clarify. But there's a slight difference in the formula between those two products. Very similar. I can't comment on concentrations, but the actual spores and different components within the product are very similar, except for one piece.
Yeah.
Talk about the difference between those. Is there a big difference in efficacy?
No. There's we've actually done a couple of small studies to bridge the gap there to see if there's a difference in efficacy in the most important functions, like leaky gut, for example, right? And modulating inflammatory responses in the gut and improving the microbiome. We don't see any measurable difference between the two in that regard. the fundamental difference is the presence of a strain called Bacillus ligniformis. Bacillus ligniformis is a really interesting strain because it produces antimicrobials in the gut to ward off certain pathogens. And that can have some die-off effect, right? And the reason why we formulated Just Thrive without it is because certain people, a certain percentage of people that take it, if you have ligniformis in there and you have pathogens, you're going to get a lot of die-off symptoms. You're going to get a lot of cramping, bloating, discomfort. It's a good sign that things are moving in the right direction. But for consumers who are not working with a practitioner, that can be alarming, right? And if you're working with a practitioner, the practitioner would have warned you and said, hey, you have SIBO, you've got a dysfunctional gut, I did a stool test, you have a lot of pathogens, you're going to get some Herxheimer or some die-off response, so let's manage it, right? So a practitioner can talk you through it. We didn't want consumers taking it and a percentage of them getting all these issues and then discontinuing it because they think it's a bad thing. So that's the main difference, is the presence of that ligniformis and the specific antimicrobials that it produces. Now, if you're somebody that's listening to this and you go, oh man, I have a lot of pathogens in my gut, maybe you've had a gut test at some point to know that, or you have any other indication, then the Megaspore would be very useful, but know that you probably will have some die-off response. In terms of maintenance and improving your gut and sealing up the leaky gut, having a healthy, you can do the Just Thrive, and that's perfectly fine, right? And in fact, that's typically what I use for maintenance myself, is the Just Thrive as well.
And are you still working with both companies?
So I've spoken for Microbiome Labs up to about the end of last year. I don't have any speaking engagements with them this year, but I do have some speaking engagements for Just Thrive as well. I do assist them with some, ideation work, and then I do some talks for them as well.
Okay. Obviously, you believe in the products and you're a scientist working with these companies, but just for full transparency.
Yeah, absolutely. I mean, I was a founder of Microbiome Labs, right? So I founded the organization and developed the technology. Was very happy to find an opportunity to sell that off to a larger biotech company who's going to take it to the next level. And then I also had other endeavors that I wanted to pursue and other problems to solve. And through that building of Microbiome Labs, we partnered with the founders of Just Thrive, who wanted to take this technology to the retail space. So that was how we partnered with them. Nonetheless, my biggest position with both of those now is I'm a customer. I actually buy the products just like anyone else off from retail sources, or if I'm buying Megaspore, I'm buying it from a clinician. I don't get any special deals or anything. The Just Thrive folks have been kind to send me a number of free bottles for helping them with some interviews and so on. But I use those every single day. My family uses it every single day. My kids use it every single day. We're now customers and users of it.
All right. Well, thank you for the conversation. We're going to link up the probiotics, your social media, your website, everything in the show notes. Thanks again.
It's my pleasure. Thank you.
Now that you're done, you're going to want to head over here and catch this next incredible episode. You don't want to miss it. I'll see you over there.
It would be hard, Jesse, to find diseases, modern diseases, that are not either initiated or exacerbated by the microbiome. Look what we're doing with reuteri, reduction of skin wrinkles, vaginal moisture.